try to do today is describe the Vigilan study. How many people here have not heard of it or don't know anything about it? Okay, a few. All right. Okay, so our goal, I'm hoping that the goal of the study will come across that we really wanted to understand the symptoms and the course of people living with CTD and contribute something to the development of therapeutic intervention programs and really think about particular endpoints. Okay, so it was an observational study. That means we didn't have an intervention. We enrolled 50 males with confirmed CTD. We focused only on males. This was also started in 2016. And the way we did the study was we had in-person visits every 6 months, and we had caregivers touch base in between those visits also. So we have a lot of surveys, a lot of updates, and a lot of in-person testing. Toward the end, we changed protocol to move to 12-month in-person visits, so that's why it says we did in-person every 6 to 12 months, but most of our data is 6-month intervals. And we had families up to participate for up to 4 years. This was originally designed as a 2-year study. It turned into a 4-year study that took 8 years, 10 years, 50 years. It seems like we've been doing this for a long time. All right, and we started collecting data in 2016. That's when we first enrolled participants, and we enrolled our 50th participant in 2020. And at that point, we kept going for another couple of years, but then it seemed like we had pretty stable data, so we did terminate data collection before everyone got their 4-year to the 4-year visit. But we have quite a bit of data. So this shows kind of how many visits we have across the 4-year span. When it says lost to follow-up, that doesn't mean that the family didn't want to participate anymore. That means that's where some of those kids, like our kids that were enrolled at the end of the study, only got maybe their 12-month visit or their 18-month visit. But most people made it through the bulk of the study. Okay, there's a little bit about our demographics and our participants, and I want to piggyback on on Dr. Curie's sample yesterday because we're trying to do similar things, right? She presented a lot of potential endpoints and collected data on a really nice participant sample. But there's differences between the two groups. So I'll try to, without remembering all the details of her study, I might draw some comparisons, and then if I'm incorrect, she can correct me. So in our study, most of our participants were young, compared to her study where I think the average was age 17, is that correct? Yeah, so the average age in the study yesterday was 17. So those kids, you'll see, were able to participate in a different way than our kids would. So about 40% of our kids were under the age of 6, and another 40% were 6 to 12. And so we're talking about a lot younger kids. You'll hear me use the word median a lot. How many people know the difference between a mean and a median? Okay, how many don't? Okay, just quickly for the people who don't. Mean means you take the average. You take everybody's score and you divide it by the number of people that you got a score on. And then you get the average. That's the most common thing people talk about. Median is where you figure out where's the halfway part. So the reason that I use median a lot here is that when you do averages, you might have an outlier. So if I'm studying a wide population, we opened this study to anybody from age 6 months to 65 years old. We didn't get that far. We only have about age 2 to 24. But if you have a lot of 3-year-olds and you throw in a couple 20-year-olds and then you try to calculate the average age, it doesn't really describe the sample very well. So median is a better, depending on what you're trying to communicate, median can sometimes be better, and that's better in our case. So our median first symptom in hindsight was 6 months. So at least half of our kids, there was an identifiable feature at 6 months in hindsight. So it wasn't what was acted upon, but that looking back, you can find something at 6 months. And half of the kids it was later than 6 months. Half of our kids had their first evaluation, not for CTD, but for something at about age 15 months. And then half of the kids, their first evaluation was later. And then there's a typo here, half of our kids had their diagnosis actually by 45 months. not 48 months. So, but some of our kids got their diagnosis in their teens, and part of that's history, because as we understand more, we can identify kids who are older who were missed early on. Another reason to use median instead of mean sometimes. Here's a little more about our participants. There's the race and ethnicity breakdown, our genetic breakdown. Our 10 of our participants had at least one sibling with CTD. There's our mother carrier status. We don't have everybody's carrier status, but that's what we know. And then that's our list of what was the first symptom in hindsight. So most of the time, you'll see that first symptom is a missed milestone or a feeding issue. And that's pretty nonspecific, but that's what we're dealing with, and that's what a lot of neurodevelopmental conditions are dealing with. So you'll see, for most people, a seizure was not the first indication. When a seizure is your first indication, that you get fast-tracked to a neurologist, and that may really prompt somebody to think about a genetic testing. But when that's not your first, there's other opportunities that we want to capitalize on. We also really tried to figure out how many people had seizures, and we broke out febrile versus non-febrile seizures. Our neurology colleagues have indicated that febrile seizures are not really going to be something that they worry about too much. They just are too nonspecific. So they wouldn't, if you came in just with a febrile seizure, they wouldn't necessarily think to get genetic testing. So we wanted to really think about how many non-febrile seizures are occurring and when. We also looked at GI symptoms. So this was in the beginning of the study and throughout the 4-year participation. Did you ever have these characteristics? And you can see the top most common characteristics are related to constipation and difficulty getting food down. Okay, here's some notable events and milestones in our population, and I think there's a lot of similarities with the data that Dr. Curie showed yesterday, but some differences. These are younger kids, so some of these kids there's still time for them to develop some of these, to reach some of these milestones, and her population is a little bit older. But in ours, all of the children were walking by age 4. 78% could use at least a single word, and those kids had gotten that single word by about age 5. However, only 34% of our sample was able to use 2 to 3 word phrases or sentences, and we didn't have really complex sentences for the most part. But when children did meet that milestone, it was between the ages of 4 and 13. Toileting is something I'm particularly interested in, and so we looked at who's really independent. So our bar was very high. You had to be fully independent day and night, no accidents, which is a pretty high bar. 18% met that, and when they did, it was between the ages of 2 and 9.5. Okay, whoops. All right, we also looked at interventions. So just here's the kinds of medications our population was on. I think everybody was taking something. I might be wrong. There might have been one person that wasn't taking anything, but I don't think so. And then there's the types of therapies that people were doing. Most people are getting at least all 4 therapies. So this is a good testament that people are able to access therapy, but this is a full-time job for families to get kids to all of these therapies. I thought it was fun how much hippotherapy was happening. And there is one kid who was not getting any therapy, and I'm not sure it's because they didn't need it. I think it's because it was not really accessible for them in their area where they lived. Okay, I'm going to come back to these in a minute, but I wanted to go over the measures that we tried and what we learned from that. So this is the list, and this is going to also sort of try to incorporate the core outcome measures that people have been hopefully hearing about. There was a poster about it. It's the work of the ACD in their grant to kind of come up with core outcome measures. So the core outcome measures related to behavior are, I have them here, adaptive skills, cognition, expressive language, fine motor skills, and emotion regulation. And you'll see these measures hit a lot of those. Dr. Curie's measures hit a lot of those. We may sometimes go broader than that in an effort to cast a wider net to make sure that we are not missing something else that could be easily measurable and associated with one of those things. But these are all examples of what you would, general cognition measures. And when kids are little, cognition measures also measure a lot of development in general. So for this study, we used the Mullen in 2016. That seemed like the right choice. It had been widely used in autism, which was a good comparison sample. And we also tried the WASI when we could. In our sample, only 4 kids could get a WASI that had valid scores. So we ended up doing the Mullen out of range for almost everyone and converted those scores to mental ages for study. What we then switched to toward the end of the trial was we thought the Bailey would make more sense. The Bailey is like the Mullen in the types of things it measures, but it has some statistical properties that the Mullen doesn't have that we can use for comparison. So they have something called a personability score or a growth scale value, which we've come to see as really, really important for this population. It helps you become your own control. So you're not being compared to every other 5-year-old, you're being compared to yourself the last time you took this measure, which is great for rare diseases and for developmental conditions. Okay, so other things that we decided, we also would swap out the Stanford Binet for the WASI because it also has this personability score. That's an IQ test that you may have seen in IEP considerations. We piloted the PPVT, which is a receptive language test, and the EVT, which is an expressive language test. Those two things are co-normed, which means you can compare them across each other with some nice, in some nice ways. We did have 32 kids could get something of a score on the PPVT, but it wasn't super robust and not everybody could do it. We didn't try it on every single participant. So there may be some promise, but that's receptive language. That's not one of the things parents are most excited about. Parents are more excited about expressive language. And we, toward the end of the study, we did have 21 participants who could get some sort of score on the EVT, so that could be potentially useful. Again, we weren't able to try it on every participant, so we might not know. There might be some other kids who could get it, but there definitely were lots of kids who could not get it. Okay, we also tried the NIH toolbox. That was too difficult, but there is a different version being validated for intellectual disability, which has a lot of the same types of activities that Dr. Curie, the tablet things were describing, so that could be promising. Okay, we also looked at questionnaires. We did the aberrant behavior checklist, which I bet parents have seen that. It's developed for kids with ID. We would use that again. We used the ADHD rating scale. We would not use that again. ADHD, we have a lot of hyperactivity, but the way the items and the ADHD criteria is not really appropriate for folks with significant intellectual disability. We also wouldn't use the parent stress index. That has been used in FDA trials, but that's really speaking about the stress between a parent and a child, not the burden of a developmental disability necessarily. So it'd be better to use something like a caregiver burden or quality of life. We also use the SCQ, Social Communication Questionnaire, as part of an autism assessment. I do think it's really important to measure autism for a clinical trial because if you have both CTD or a CCDS and autism, that's different than if you just have one without autism, because autism may contribute its own challenges to treatment and to other kinds of things. And then finally, we used a clinical global impression scale, which we would use again, but we would create more anchors and more training. We also used the DDC-GAS, which is another clinician rating scale. And I put these up just to let you know, we did echocardiogram, we got pretty good data on that. We tried to use a wrist embrace wearable to track seizures. We did not have success with that. That might be because our kids were so much younger than Dr. Curie's kids. And then we did try to get ERPs and EEGs with a cognition cap that was designed with very few leads. It's supposed to be very portable. You can just snap it over the head and bang in some gel and it measures really fast. A lot of our kids did not enjoy wearing that. You know, it takes about 2 seconds to take that thing off. We did find they got better at it with visits, but it was not robust enough where we felt like we could really get get everyone to participate. And in the end, the company didn't have child comparison data when we needed it, so we wouldn't use that again. But there's maybe other things on the horizon. Okay, so I wanted to make sure you knew about the measures because measures are important. And then let me show you what we found. Okay, yesterday, Dr. Curie talked about kids being on the floor. Here's a picture of what that means to be on the floor. This is the Vineland, which is an adaptive test, and these are the scores that our population got. So you can see how there's so many kids at the bottom. That means that the test didn't have items that were really low enough to really capture their ability. So they end up on what looks like the floor. And so what's bad about that is that we don't know how far down off the floor they are. So if we have an intervention and it makes a difference, it still may not be enough to show because it may not be enough to get them off the floor. So what's a better measure is when you have something called a growth scale value or a personability score, like I was mentioning, where you can compare yourself to yourself in the past. Unfortunately, you can't convert an old test into something with a personability score. It has to be built in from the beginning. There's more and more tests that do this, but they don't all do that. The new Vineland does do this, and you can see how the kids are not on the floor, right? So there's several areas where they're not on the floor. The red line is where you would expect scores to be if there were no developmental concerns, and you could see our participants are remaining kind of in that impaired range, but there is some movement, and we did find that they do move, which was great. Let me see where. Okay. So two things that we're going to put in the manuscript, which again is being currently reviewed by the authors at the moment, which is great. Kristen Farmer did analyses for us at the NIH, and she tried to think about how can we use the data we have to model what would you expect in a different sample if our sample holds for other, can be generalized to other CTD populations. So without going into too much detail how to do this, you'll see this dotted line again is where you would expect kids to be if there were no developmental concerns, and you can see our participants are all the circles, and then there's gray lines in between that are kind of like shaded. That's each of their regression. And then she took all that and she created an estimate, a model estimate on the right-hand side that shows what you would expect based on our data, how you could model that for future samples. And so what you'll see here is in some areas, you would expect different amounts and lower amounts of change depending on the child's age. It's not linear. It doesn't mean that that as you get older, you get less ability to change. We don't have the data to confirm that. What we have is, in our sample, the kids who were older, it would suggest that they would show less change, but not in every area. So coping, look at that. Everybody's learning how to cope pretty good. You can still learn lots of good coping skills, which is great. But I think this will be really important as we think about clinical trials. What do we expect at different ages? You usually start clinical trials in older populations, and so knowing what to expect and finding a measure that could be sensitive to that is going to be really important. Okay. All right, let's stop there. It's 10:30. And do we have any questions? Okay.
Hi, Judith.
Hi.
Hi, Judith. That was a really excellent talk. I think a lot of hope, actually, that you can get it to into the grass to measure change in where, to recognize tools that are not helping you with that, and to really define and refine the tools that will help the clinical trials, because any benefit for a treatment is worth measuring. Does anyone in the audience? Yes, thank you.
Thank you. Wonderful talk. So in preparation of, let's say, clinical trial, I see a talk was there from yesterday from Dr. Curie, and I saw wonderful data from you. can this data be combined, and can we come up with a list of tests which will be more relevant and can be done, let's say, in Europe and in North America
Right.
together?
Yeah, I think it's a great question. I think we might be hitting a nice point in as a community where there are enough studies and enough different populations that you can find different things. Like when there's only one study, then you assume that that's ground truth. So I think it's great that we have a couple different options. And yeah, I think there's a lot that we can learn from each other on what worked in our different age ranges, and especially thinking again about what would be sensitive at different ages. I mean, the goal is to try to get treatment in as early as possible. So there's a need for kind of everything. There's a need for like what would be measurable very early on, but also what would be measurable in an older person who's more likely to be part of the first clinical trials. So yeah, I think, and the Europe and US and different measures and different norms, all of that there's room for understanding more how to do it. I think it means, I mean, we can combine what we've done, but we also probably need more natural history of older kids. We didn't have, our sample is quite younger, so there would be a benefit for having more natural history of older kids too.
Oh, yeah, thanks.
Thank you. That was a lot of effort that you guys put in. In terms of the measurements, I see that there are a whole list of different measurements, and you guys changed it, some of them in the middle. So if when you change it, there's no baseline measurement, so how did you mitigate that? And then did you see any measurements that you guys used that showed, I guess, good progress within CTD patients that could be used for clinical trials?
Yeah, great question. So the best measure we have is the Vineland, because it covers a lot of self-help skills, which hits a lot of functional communication and daily living skills, and it has this growth scale value that you can use over time. And in this study, we happened to be able to, it came out right in the beginning of the study, so we were able to use that throughout. For the things that we pivoted, once we pivoted, we made the decision that we may not be able to say much about this long term. We're going to do it as a feasibility. Can we get data from the kids? So sometimes we only got one visit with that measure, and the goal really is, we already had learned what we could from the measures up to that point, and we wanted to take the opportunity to see how feasible that measure would be in administering it. Great.
Thank you. Are there any more questions?
All right.
Okay, I think in the interest of time, we'll...
Yeah. Sounds good.